Tomorrow there'll be the CAO Seminar at 11 o'clock in room 102. Itamar Kalbi from Weizmann will be talking about reassessing the future changes in mid-latitude atmospheric waves. On Sunday, geological survey at 10:30. Noy Levy will be talking about condensation and hydrated mineral recycling in Dead Sea brine at the solution area. And that's it. Those are the two seminars for the coming week. Today there will not be an almost weekend meeting after the seminar, because there will be a faculty meeting instead, but there will be 60 seconds, won't there? And so now I'll hand things over to Adi, and he'll introduce us.
אוקיי, אני אתחיל כמה מילים בעברית, נראה לי שכולם פה. אז אני מאוד מאוד שמח להציג את נוי לוי, תלמידת דוקטורט, זה בעצם הרצאת הדוקטורט שלה, שהיא ביצעה בהנחייתי ובהנחיית רלף שיבל ממקס פלנק בגרמניה. ונוי היא סוג של תוצר קלאסי של הקורונה. אני לא חושב שיש הרבה מקרים שתלמידה ומנחה נפגשים בפעם הראשונה שנתיים בתוך הדוקטורט, וזה מה שקרה לנו. אני הייתי בשבתון, ובדיוק הקורונה תקפה אותנו, ונוי הייתה בגרמניה, והכל השתלב בצורה נפלאה ונהדרת, וגם העבודה הייתה נהדרת, אבל נפגשנו רק אחרי שנתיים, אז זה המצב. ואני מאוד שמח שהגענו לרגע הזה. אני אעבור לאנגלית עכשיו, אז so I'm happy to introduce Noy Levy. Noy is giving her final presentation for her PhD. She did undergrad in biology and ecology, and then went on to do a master's in scientific education in Be'er Sheva, after which she moved to this topic in the Hebrew University with Max Planck Mainz, working on the geochemical proxies in Planktic Foraminifera. And essentially the work Noy has done has been groundbreaking in the sense that she's taken it to the next level. For those of you familiar or not familiar, forams in paleoceanography and oceanography are central tool, but we always look at them as a whole. What Noy has done was to take them apart, these tiny beings have been taken even further apart to minute analysis, and this is the work Noy will be presenting. So, Noy,
so first of all, thank you for being here. And today I'll present my PhD work on calibrating geochemical proxies using Planktic Foraminifera from the Gulf of Aqaba, Northern Red Sea. So yes, my advisors are Adi from here, the Hebrew University, and Ralph from Max Planck Institute for Chemistry in Mainz. I'm very excited, so nervous, please forgive me. And before diving into the geochemical part, I want to give you a brief introduction to modern Planktic Foraminifera in general. So they are single-celled marine organisms. They have a calcareous shell. They're found globally, and some of them have symbionts, so these are microscopic algae that either live inside the shell or on the shell, and both the foram and the symbiont enjoy this mutual beneficial relationship. But most importantly, they are not uniformly distributed in the ocean. So some of them live in the surface mixed layer, some species near the thermocline or underneath the thermocline, with other groups are found in high latitudes and high productivity areas. So this ecological partitioning is important because Planktic Foraminifera calcify their shell from the water in which they grow. So the chemical composition of it, the proxy systematics, is basically habitat specific and not some average ocean signal. Furthermore, they calcify their shell one chamber at a time. So let's try and use this. You see here the final chamber marked with F or F0 is the last one to calcify, and then the F-1 was calcified before it, and so on. So this gives us a timeline of their life cycle, or the whole story, basically. So, as I said, Planktic Foraminifera calcify their shell from the water in which they grow. So, when they do that, they record the water column conditions such as temperature, salinity, pH, and other hydrological parameters. But this record is affected by the species ecology as well as calcification mechanisms. This is why calibration studies are still very much important, especially in regions that proxy systematics are poorly constrained, such as the high salinity Gulf of Aqaba. In this study, the main premise is that we can use high resolution time series analysis to shed light or better understand proxy systematics in the Gulf of Aqaba. So I just want to give examples to some of the proxies. So we have chemical proxies. So we use, to reconstruct seawater temperatures, for example, magnesium-calcium ratio or delta O18. We use sodium to calcium for salinity, boron to calcium for pH, delta C13, trophic level and DAC, barium to calcium for phytoplankton productivity. We also have ecological proxies, such as species assemblage composition, diversity, shell sizes. Of course, there are more. I'm not listing all of them here, but these measurable parameters allow us to gain information on the water column. So I want to show an example of the magnesium-calcium ratio as a thermometer. So magnesium incorporates incorporation to the shell calcite increases with the increase of temperatures. And you can see here in these two bottom panels that there's this positive relationship between magnesium-calcium and annual sea surface temperature and seasonal sea surface temperatures. But we can also see here that this relationship differs in between species. So it is species specific, and each species will require a different calibration for this thermometer. If we move to the right-hand side of this slide, we can see the magnesium-calcium thermometer in action. So this is an example from a Pliocene record from site ODP 806. And I want you to look at the orange triangle is the modern sea surface temperatures. And if you look at the orange curve, you can see magnesium-calcium ratios from Saculifer. This is a species called Saculifer. And we can see that this is a very powerful record that it reconstructed temperatures in very high resolution all the way to 5 million years ago. Saying that, we see that if we look at the black curve, the TEX86, which is another thermometer, in some parts of the record, they are not, they don't align perfectly. So this is another example why we still need to do this tweaking and calibration studies to better understand this tool. So I started with the introduction and motivation, and I'll continue to talk about the methods, and I will show the first two chapters of my PhD, which are published papers, and I'll move to the third chapter, which is not yet published, and then I'll finish with synthesis and conclusion. So the study area, the Gulf of Aqaba is located in a hyper-arid climate area, and also it is oligotrophic, meaning it's poor in nutrient. There's very minimal river influx. Salinities are very high, and it is evaporative driven, around 40.5, 40.7 salinity points. And the temperatures, see here, there are two layers to the water column. So the bottom layer is constant, around 21 degrees Celsius, and the seasonality in the surface layer with a thermocline, a relatively strong thermocline, that with the cooling of sea surface water, it deepens, the thermocline erodes, and it leads to water column mixing even up to several hundred meters. And specifically, in the time of this study, it was during the March till May months, so I call them the water column mixing months. And the Gulf of Aqaba is an excellent site to carry out studies like this because it builds on an extensive body of knowledge provided by the National Monitoring Program. So we have more than 30 years of hydrological data that allows us to compare hydrological data measured real time with shell geochemistry. So let's move on to the Planktic Foraminifera. In the Gulf of Aqaba, there are 10 main species. I list them here. You can see their shorter names here, and you can look at them here. These are SEM images. And the first, the most important is that Planktic Foraminifera flux is seasonal. So during summer, fluxes are very low, and during winter and water column mixing, Planktic Foraminifera flux is much higher. And important observation from Natalie Chernichovsky's work from 2018 is that the total flux, you can see it here in the upper panel, panel A, the total flux of the Planktic Foraminifera is dominated by the smaller size fraction, the 63 to 125 microns. And this size fraction here, if you look at panel B, you see is dominated by a species called T. clerkii. After that, the 125 to 500 microns fraction size is dominated, here in red, by a species called G. ruber or G. ruber albus, because there's also a pink variant was not found in the Gulf of Aqaba. So an advancement of this study is that we utilize the smaller size fraction and we measure it as well because, and it is very important because if we would have ignored the smaller size fraction, we would have ignored the major part of the export signal. So my PhD objectives follow three simple questions. The first question is, can we use the magnesium-calcium ratio in hypersaline regions, or is it fundamentally broken because of the high salinity? The second question is, what is the interchamber variability, or what is the proxy systematics between chambers in the Gulf of Aqaba? And can we use T. clerkii, which is a species we hardly know anything about, as a potential paleoproxy? So all the material for this, the sediment, the Planktic Foraminifera material was collected from, for this research was collected from Station A. So a bottom-tethered mooring station was deployed near Station A, and it was vertically stacked with five sediment traps in 5 different water depths. Those sediment traps were retrieved on an approximately monthly resolution, and we also sampled the core top sample from Station A. And this setting allows us to compare and to investigate shell geochemistry, proxy systematics with time, because of the different months, in the water column, as well as post-depositional preservation in the same natural system, and to compare it to real measured hydrological parameters. So I focus on the 2 flux-dominating species. This is G. ruber, and this is T. clerkii. And these 2 species, they represent 2 different lifestyles. So G. ruber is a surface dweller. It resides in the mixed layer, and it has symbionts, while T. clerkii is the smaller fraction. It is symbiont barren, and it is presumed to dwell near the thermocline or underneath the thermocline, but saying that, we hardly know anything about its biology. So this setup allows us to first compare these two different layers in the water column in terms of proxy systematics, but also hopefully shed light on the poorly constrained T. clerkii. Furthermore, in the Gulf of Aqaba, T. clerkii is appearing in two different phenotypes. This one is the big phenotype, which all of its chambers are fully recognizable, and encrusted phenotype, which its shell is a bit thicker, and the chambers are a bit harder to distinguish. And I'm giving you here a bit of a glimpse forward. You can see the chamber-specific resolved measurements that we've carried out. So at the MPIC in Mainz, I measured single-chamber trace elements measurements using laser ablation ICP-MS. The spot size that was decided was 30 microns. The reason why we went with this spot size and not larger, for example, is because this one was the largest to fit one chamber of the smaller T. clerkii. We intentionally avoided oxidative and reductive cleaning because it would, we thought that it would mask the natural signal of the trace elements, as well as the T. clerkii shells are very delicate and it could have broken them. Yeah, sorry, and of course, these are the set of elements that are measured, of course, sorry. An important point that is important for this paper, this work, is that work, the fit was not good. So we moved on to create a local exponential calibration for GeoRuber. It is valid for only high salinity, so about 40 to 41. But although we call it local calibration, in theory you can use it and apply it to other marine environments which also have very high salinity, either modern or paleo. And an important note that I want to make about this is that until now, high values of magnesium, calcium, they occurred before in other places, but they were either considered, they were either excluded from global calibrations or not just global, from the calibration, or was treated as an anomaly. And my main point here is that this is real data, and these high magnesium calcium values belong inside the calibration frame, and therefore we revised a global calibration from Gray et al. From 2018, and we added to that our Gulf of Aqaba data, and Metzger et al. From 2018 from the Red Sea data, and extended this global framework to account for higher salinities. I understand that this calibration is not the strongest or most robust, but I believe that if we add more data to it, it will improve its strength. So a take-home message: magnesium calcium of GeoRuber can be used as a temperature proxy, but we must take salinity into consideration. The second paper builds directly on the first, but it is much broader in scope. So here I look not only on the magnesium-calcium ratio, so I look at the entire set of trace elements, as well as I look at proxy systematics between chambers. So here I show you pooled means of the entire set. As you can see, it's a lot, so I'm not going to discuss all of them. I'm just going to give some examples. Okay, so what we see here are pooled means of each sediment trap. So each sediment trap has a different icon, and each mean stands for the entire set, year, so the entire year, and all the chambers as well. Also, we measure I show here the core top values. So pooled means are species specific and element specific. And what I mean by that, I'll explain it with an example. If we look at the barium to calcium concentration, for example, the pooled means of GeoRuber, they fall under a small range. Compared to GeoRuber, thick Lerchii, the pooled means of thick Lerchii, both big and encrusted, they have a much larger range of values. If we look at iron to calcium, for example, if you can see, the range of pooled means is very small and very similar between the three species. So it is both element specific and species specific. Another important observation is that core top values sometimes differ very much from the water column samples, like we see here in iron and in titanium to calcium. We see it a lot in the lithogenic elements. So this is very important for reconstruction studies, because when we go to the core and measure our samples, we want to know that the values that the data that we get actually stands for water column conditions and not other mechanisms that could have been taken place. Let's dive deeper to the chamber level. So we see here the three species: GeoRuber on the left, thick Lerchii encrusted on the right, and big in the middle. This is MLD depth in meters, and this is magnesium calcium. And we see each color of different curves is a different chamber. And what we see, again, is the rise in interchamber variability and higher values during water column mixing months. And also we see that there are more interchamber variability also between sediment traps, as we can see it by the length of the error bars. But my point here is that this observation is not unique to magnesium calcium. We can also see it in boron to calcium, in barium to calcium, in thorium to calcium, and uranium to calcium. So, and this is very interesting. So this water column mixing anomaly hampering the ability to recognize perhaps seasonal trends, but it opens the door to use mostly T. Clerkii as a proxy for water column mixing, for water column stability. I said that it hampers our ability to track seasonality, but it is not the case for magnesium, calcium, and boron to calcium in G. Ruber. We can still trace this seasonality, and in scarcer element we see the noise is much stronger, so much more extreme values and stronger fluctuations. Another observation is that the final chamber, if you remember it here, it has a different behavior to the previous chambers. Again, in several trace elements, not just magnesium, calcium. So in G. Ruber, the final chamber is the values of the final chamber tend to be lower. You see it here with the orange, lower than the previous chambers. Whereas in T. Clerkii big and encrusted, it's usually higher than the previous chambers. And the question that rises from that is why? Do they have different calcification mechanisms? So it's an open question. And another very important observation is that T. Clerkii has more interchamber variability compared to G. Ruber. And again, when we ask why, we can explain it in two different approaches. Either is that they do have different calcification mechanisms, so T. Clerkii is more efficient in incorporating trace elements to its shell, or we can explain it ecologically. T. Clerkii dwells underneath the thermocline. It is exposed to a more heterogeneous environment, richer in nutrient, compared to G. Ruber, who dwells in the surface layer, which is more homogeneous and more nutrient depleted. So it could be both. The most practical outcome of this paper is that we used the local calibration from the first paper on a chamber level. So here, what I'm showing you here are calculated temperatures from the magnesium calcium for each sediment trap. You can see it here, the different sediment traps, and compare them to measured temperatures of the MLD and the respective depth. And what we see is, first of all, we see that there is only good fit between calculated temperatures and measured temperatures in the upper sediment trap. For G. Ruber, of course, sorry I didn't say it, which makes sense. Again, G. Ruber calcified its shell in the upper water column, so these will be the conditions that it records. But interestingly, while the F-1 chamber and F-2 chamber show a good fit with measured temperatures, the final chamber does not. And therefore, we suggest when using magnesium calcium as a thermometer to use non-final chamber pooled means. So to conclude this chapter, proxy systematics are species-specific, element-specific, chamber-specific. Interchamber variability could, in T. clerkii, can be masked, could mask environmental and seasonal signal. But we can use T. clerkii as a recorder of water column mixing, and we should always check and compare core top values to water column values. So the final chapter of my PhD asks, can we use T. clerkii for paleo reconstructions? Again, this species is the most abundant species in the Gulf of Aqaba and probably in other locations, but still we know very, very little about its ecology. Again, its ecology matters because this is the lens in which we interpret the geochemical proxy. So let's continue. So looking at its global distribution, we see that this species is actually quite tolerant to a wide range of temperatures and salinities. So between 8 degrees Celsius to 30 degrees Celsius and 32 to 41 salinity points. And it is very dominant in many areas. Everything I'll show you now is new. We don't know anything about it, okay? Except for its fluxes in the Gulf of Aqaba, which of course Natalie has done a great work. So here in this chapter I'll be examining its morphology, the trace element to calcium ratio I studied in the second chapter, and I also add isotopic composition. Okay, so using light microscopes, so binoculars, SEM, and nanosims measurements, I investigated its wall structure. So after imaging more than 2,000 individuals, I can say that there is a significant difference in the size between the big and encrusted. So the big is indeed bigger. And when it comes to aspect ratio, the big phenotype is rounder, while the encrusted type has more of an egg-shaped. Furthermore, the big phenotype has only one layer to its shell, as you can see here, from the SEM, while the encrusted type has 2 layers. I don't know if you can see here the SIM, but it has 2 layers. Also, from nanosims measurements, we see that the chemical composition of these 2 layers is different. If you look here at panel 4, you see that the interior layer, the inner layer, is more calcium depleted than the exterior layer. And something that also is very, very interesting is that it calcifies its exterior layer while it is still alive. And we know that because of the pores, they're open. If you can see it here, they're open in both layers. And how does he uses these pores? So he sends cytoplasm shoots to collect food from the outside and bring it inside the shell. So it calcifies while it's still alive. And this strengthens our hypothesis that this exterior layer is most likely a gametogenic layer, which produced after reproduction. So our belief is that the big type and the encrusted types are 2 phenotypes of 2 life stages of this forum, before reproduction and after reproduction. So I plotted the encrusted to big abundance ratio with time, and I saw that there's a very nice trend, and that this ratio is higher with higher temperatures and lower with lower temperatures. So I want to suggest that although the magnesium-calcium ratio is not good in T-Clercii to reconstruct temperatures, maybe we can use this phenotype ratio as a new proxy for temperatures. And I also measured isotope composition, stable isotope composition, so delta O18 and delta C13. So just a quick reminder, and we measure the delta O18 of the shell in order to gain insight on the calcification temperatures. So if we know the temperatures in which they calcify, hopefully we'll know the depth in which they calcify, so their depth habitat. And the delta O18 of the shell is affected by the delta O18 of the water, salinity, temperatures, and possibly vital effects. But what I want to say quickly is that higher calcification temperatures will lead to lower delta O18, and lower calcification temperatures will lead to higher delta O18. And here I show a monthly resolution of both G. ruber albus in red and T. Clercii big in light blue and purple is encrusted. I added here the G. ruber for comparison because we know more about it, and then we can maybe understand something about the T. Clercii. But also there are two theoretical forums. One that calcified its shell in the temperatures of the 450 water depth, meters water depth, and one that calcified its shell in the temperatures of the surface water. And if I look at the G. ruber in red, I see that there's seasonality. It goes very well with the surface theoretical forum, but T. Clercii is very interesting. What's going on here? It has much lighter values compared to G. ruber, and even the surface, the theoretical surface species. And although it shows this nice trend, we would have expected it to be much heavier because it calcified shell underneath the thermocline, not in the surface. So we think that it could be either vital effects or rapid calcification that causes a kinetic isotopic fractionation. But I want to say that this although it is an amazing observation, it's still unresolved. As for the delta C13, we measure delta C13 to understand what it eats, basically. And so the photosynthesis causes delta C13 values to be higher, and respiration makes the delta C13 values lower. And again, I'm comparing the T. Clercii with G. ruber, and we can see here G. ruber is in green, and T. Clercii big in orange, and encrusted is in blue. And we see a great differentiation between G. ruber and T. Clercii. G. ruber again is a symbiont bearing, and it is carnivorous as well, but it's in the surface water. And this trend aligns with a species that is detrivorous, that is feeding on the export material underneath the pycnoc line. This is very low. So we think it is a detrivorous species. So to conclude the final chapter, although this is from the previous trace element to calcium, ratios in T. Clercii could infer water column stability or mixing. The two phenotypes of T. Clercii likely represent two different stages of this species. before reproduction and after reproduction. The encrusted big ratio, phenotypic ratio, is suggested here for the first time as a temperature proxy. And T. Klierkii delta O18 is still unresolved, but from the delta C13, we suggest a deltrivorous lifestyle. And my take-home message to you is that use T. Klierkii as a proxy for paleo studies. And to summarize the entire work, the magnesium, calcium, and the delta O18 of G. Ruber are good proxies for temperatures, even under high salinity conditions. Proxy systematics should be interpreted with interchamber variability, and I propose here the T. Klierkii as a new proxy. And if you want to be more practical, I suggest preferring region-specific calibrations and be specific when you choose your chambers, and explain interchamber variability when you reconstruct when you use it in reconstruction studies. And in that note, I want to thank you all for listening, and I want to thank my advisors, Adi and Ralph, and my committee members, Boaz Lazar and Sigal Abramovich, all my collaborators and co-authors, Natalie Chernichovsky, Klaus-Peter Jochum, Ulrika Weiss, Brigitte Stroll, Jan Leitner, Sonia Shaban, Hubert Vonoff, and Gerald Howe. I've put a picture of Klaus-Peter because he sadly passed away during this PhD, and I wanted to honor him and to acknowledge his tremendous contribution to the trace element measurements and the laser ablation ICP-MS. I want to thank DIY Marine Crew and Barak Yarden, and of course my husband and kids and family and friends. That's it.
Good. Questions? I have no questions.
With regard to the laser ablation analysis, is it possible that during the ablation you sampled a neighboring cell, or you sampled different cells, yeah? Is it
Chambers.
possible that under the surface you sampled or ablated a neighboring cell, and if yes, how do you recognize it, and what did you do about it?
Okay, I don't believe I sampled different chambers as well. The ablation dwelling time was about it actually changed between the species. G. Ruber has a much thicker shell compared to T. Klierkii. But when we look at the profiles, there's a peak during the shell, and then it goes down again. And if we would have measured another chamber, it has its shell, then it would have shown another peak, and that I didn't see that. If it makes sense. Yes. Yeah. Okay. Okay.
Thanks for the very nice talk, nice talk. And you emphasized the importance of the surface mixed layer, but at least for the smaller species, you also said that it lives below the surface, so the temperature change will be small, and yes, the main change between seasons will be the productivity and maybe something related to the reproduction cycle. So how does productivity of feeding relates to magnesium?
More nutrients, and then maybe rapid calcification, or just exposed to more elements that it can incorporate to the shell. It's not my line of expertise, but I would suggest something like that. Okay, but it could be actually also temperatures, and it could be that it is very sensitive to temperatures, and just because it is exposed mostly to very constant temperatures, we don't see the changes, and when there is a change in temperatures, it actually shows a very not extreme, but a very good reaction to that as well. So it's not that it's not sensitive, it's just that usually it is exposed to very controlled conditions, and then there's something changed. But I cannot put my finger on exactly what process was the main cause for that change.
So I have a question about the encrusted samples or species. So you mentioned that it relates to reproduction, post-reproduction. Why? What's the connection?
Some species, not all species, for example, G. ruber does not show a gametogenic interior layer. After reproduction, they go deeper in the water column. So they calcify this exterior layer in order to preserve the shell from dissolution.
Any more questions? Okay, so let's thank you.